Welcome back to our class, guys. Today we're gonna be talking about the memorandum of paper 2, so that we see how did we manage to score high marks on this paper. All right, without wasting time, I hope you guys enjoyed the paper. Yes, you enjoyed the paper. If we look at our predictions and the discussion which we did, we really covered 99% of the paper, meaning that whatever was set in the paper, we covered it. And if you go back and play the video which we did our last minute, you'll find out that yes, we have covered each and everything. Let's go to the paper without wasting time. It is M.Saidi. Thank you so much, guys, for subscribing, for liking everything. Yes, let me know if you have a question. It is a live class. I will be able to sort out that question you are talking about. All right, the first question is saying that they are saying that which one of the following is a characteristic of African apes and human in common, meaning that which of the two is in common? We say it is opposable thumb, meaning that we said it, you remember that. So 2 marks there. Meiosis is biologically important. We say that to produce haploid gametes. We said it, we look for it. Where is it? It's here. So we get 2 marks there. We're saying that reproductive isolation characterized by animal displaying specific action to attract the mating pattern. Remember, even I gave you an example that except the humans, they like to copy, and then they say that, okay, we call it species specific courtship behavior. And I said that the way how you do your dating is not the same way how frogs, how dogs, how cats, and I said that except but humans, they like to copy. Yes, all right, so this one becomes the most correct answer. So we go to the next one. Then they're saying that which one of the following genotype is homozygous, dominant, homozygous. So I need to look for letters which are the same, but they said dominant. So this one is dominant, this one is recessive. So it is C. So C becomes the most correct answer. They're saying that how many alleles control blood groups have been singing this song? We say there are 3: allele for A, allele for B, and allele for I, allele for O. So we said it, 2 marks there. Guys, the paper was flowing. Okay, which one of the following is an example of discontinuous variation? We say that anything which you are born with and it's exactly the same way it is. We said blood group. We say the height is different, the skin color. You are born yellow bones, now you are dark skinned like me. Color, the hair color keep on changing. You understand? So the answer here is B only. If you are blood group A, you are born blood group A, you will die blood group A. So that becomes the answer. So 2 marks there. Don't get disappointed if you see that your answer they are not like the same. Yeah, still at least if we get maybe 80 out of 100, maybe 80%, that's a distinction. Yes, so to fall in a distinction doesn't mean that you have to get 100%. No, it's 80%. All right, we continue. Which one of the following is a correct description of stem cells? Stem cells, remember that we say that these are cells which are unspecialized. They have ability to divide and form specialized cells, meaning that they have specific function. So we have to look for that. So we have to look for cells, specialized cell that divide by mitosis, divide by mitosis to produce cells that become specialized for specific function. So B becomes the most correct answer. We go to the next question. We are saying that a list of different lines of evidence to support the evolution given by we say that the 3 evidences, we say that fossil evidence, genetic evidence, cultural evidence. I don't need even to read the rest. Those are the 3 we talked about. So we look for it. 234. 234. 234 is C only. So we go to the next question. Say that the police arrested 2 suspects accused of housebreaking. Their DNA profile was analyzed and compared to the profile DNA. Okay, let's collect it from the crime scene. So they're saying that what conclusion can be made from the DNA analysis conducted by the police? So we need to look at, are they corresponding? No. They are not corresponding. No, they are not corresponding. No, they are not corresponding. No, they are not corresponding. It looks, but still this one is a little bit up. No, they are not corresponding. No, they are not corresponding. What corresponds here? Nothing. Only this one corresponds, but this one no. This one corresponds, but this one no. The answer is no, meaning that there is no suspect. The suspect here, no one who did that. So the answer here we have to look for neither of the suspect are positively linked to the crime. So D becomes the most correct answer, because there's nothing. If they were matching, they're not matching. Therefore the answer becomes none there. So those 2 marks. So we got the next question, the terminologies. I hope guys you got 18 out of 18. If you got 18 out of 18 or 14 out of 18, congratulations, clap your hands. All right, we got the next question. Alternative form of a gene, we say this allele. Allele, DNA containing organelles found only in plants. Chloroplast. Chloroplast. Yes, they are saying that the diagram representation showing the possible evolutionary relationship among the different species. We call it what? Call it phylogenetic tree. Yeah, we call it phylogenetic tree. Allele that is masked in the phenotype, masked meaning that if we say that if the vice president is there, if the president is there, the vice president cannot talk, meaning that there is that allele which is being masked. So we call it a recessive allele. So we say recessive allele. They're saying that the permanent disappearance of species, we call it extinction. The deliberate breeding of organisms for desirable characteristic, we call it artificial selection or selective breeding. Artificial selection. They're saying that genetic disorder characterized by absence of blood clotting factor. We said it. Then we did a lot of examples here. Hemophilia. Someone is gonna say, does spelling count? It counts. A group of organisms of the same species, that habit. A group of organisms of the same species in a specific habitat at a particular time. How do you call that? We say that it is called a population. We say that, hey guys, give me happiness. I told you that when I come back here, I need happiness. Yes. Chromosomes pair in 1 to 2. We call it autosomes. There's nothing we didn't do, guys. There is nothing we didn't do. A representation of number, shape, and the arrangement of all chromosomes in the nucleus. The moment you see the nucleus, that a nucleus we say is called the karyo. So it's a karyotype. We talked about it. All right, they're saying DNA containing material in the nucleus of a non-living DNA. How do we call that? We say that we call it chromatin network. Chromatin network. Before the DNA divides, before the cell divides, it is chromatin network. Yes. Because that's why they said a non-dividing cell. So it's B only. They're saying that location of DNA. Where do we find DNA? We find in a nucleus, in the chloroplast, and the mitochondrion. So the answer is both A and B. We continue. They're saying that the first Yo, alhamdulillah. People say 3 out of 10. No, don't. Others saying 10 out of 10. Don't cry, my man. Who is there? Zanele. And then who is that? Guys, Kahiso. All right, guys. Tabom. All right. We continue. They're saying that Hey, by the way, Alhamdulillah means praise to God. Yes. You're saying that the phase between two consecutive cells, there is nothing here. Remember, it's called a growth phase. Inter in between the two. You understand? That's why I said use IPMET. Interphase means in between the two phases, the two growth, the first growth and the second growth. That's it, interphase. So the answer here is none. 2 marks. We go. We kill the paper. We said we're gonna smash the paper. The diagram below represents two biological processes. So if you look at this, we see this is transcription. Why? Because you are forming a messenger. Check U here and then check T here. So that is transcription. And then if you look at this is T, this is T, T meaning this is DNA replication. So okay, there are some questions I asked you to do. Diagram 1, transcription, we said. Diagram 2 is DNA replication. Identify X. What is X? If you look at X, where is X? X is a messenger RNA. They are looking for this molecule. It's messenger RNA. This is messenger RNA. So correct. Someone saying that we'll try next year. For us, we will be here always to help you. We will be here, God willing, helping you so that you get what you want. Yes. So keep yourself on THUNDEREDUC and other channels, Facebook, TikTok. Yes, we are always there. Yes, but there are some people who are having different channels of THUNDEREDUC on Facebook. Those people, there are some people who have opened. So guys, please watch out. They are asking money from people. Please make sure that you don't become a victim of sending money to people who are requesting you. They pretend to be THUNDEREDUC. Yes. So we continue. If you are going to send the money to THUNDEREDUC, for example, you are buying a book, it must be an account which is linked to THUNDEREDUC, no e-wallet. Yes, all right, thank you. We continue. We're saying that identify the molecule X. So we done that with that molecule X. What is molecule X? Is messenger RNA. We say this is the messenger RNA. Natural shape of Y, we talked about it, and I said only one answer. Natural shape of this DNA is double helix. Double helix, nothing else. They're saying that the phase of cell cycle during which the process in diagram 2 occurs. Diagram 2, these are the questions I asked you. We said it's interphase. When does it occur? It occurs during interphase. Full name of the nitrogenous base that is found on molecule X, not Y. Molecule X, meaning that it is found on messenger RNA but is not found in the so the answer here is uracil. They're saying name the sugar found on X. Sugar found on X is the RNA, therefore it is ribose sugar. It's ribose sugar. Someone is telling that, hey sir, write nicely. Sometimes also when we are marking you is the same thing, you don't write nice. All right, bond Z. Obviously when they ask you bond Z is this one, is this one. So it is weak hydrogen bonds. Weak, I told you in the word weak must be there. Hydrogen bond. Yes, please make sure that is there. They're saying state the number of codons. We say that codons are three bases on X, so we're gonna count them. How many codons are there? This is 1, this is 2, and that is 3. So I see, did I say on X? No man, that is not X. Let's check properly. On X, we are seeing it here. It is 1, it is 2, it is 3, it is 4. So it's 4 bonds. So the answer here is 4. They're saying that not 4 bonds, 4 codons. Then we continue to the next question. 1.5. We go to 1.5. In a plant species, gene coding for plant height has 2 alleles. The allele for tall is capital T, the dominant over allele for dwarf, which is small t. The gene for flower color has 2 alleles, where allele for red, okay, and then allele for is dominant over allele for white, which is small r. Dwarf and white plant is crossed with dwarf and white, okay, crossed with homozygous tall, meaning is like that, crossed with the heterozygous red, meaning that red, remember, is heterozygous. Okay, state the characteristics that is being investigated in this. So we have height. First of all, we have that, and then someone is gonna say height. Okay, cool. And then we have height, and then we have, they said flower color, flower color. Cool. They're saying that give the genotype of the plant that is homozygous tall and heterozygous red. Give the genotype homozygous tall. They said homozygous tall and heterozygous red. So it's like that. So meaning that you get a mark there, you get a mark there. Below is incomplete Punnett square. Okay, we say that what is the genotype of X? I will just say, remember this goes with that. So I'm gonna say capital T, small t, capital R, small r. Get that 2 marks. A is 1 mark. Then they are saying that the phenotype for Y, meaning that if the phenotype for Y, I will need to know what is Y. So it's gonna be capital T, small t, small r, small r. Capital T is tall and white, because small r is white and white. So it's gonna be tall and white. This is the answer. I was just using this to identify what is it. What is this Y? So it is tall and white. And then this one, you get that mark. That is dihybrid cross. I told you it won't be a big problem. This one, the way we set it, the way we stitch it is different from the way we set it. It's simple. That question is done. We go to the next question. Don't tell me that you didn't do this, because we repeated it several times. Diagram 1 and 2 show the cells of the same organisms in different phases. They are not on scale. I told you, when you see it, these spindles must be in, look, in these sides, facing that side. And I told you that when this diagram comes, what should you do? Just rotate your paper, just rotate your paper to look like this, so that now these things, these spindle fibers and the centrioles, they are so don't you see that there is a proper space in between, which shows that this is the anaphase. And then even this one, you see that there is a proper space. This is also anaphase. So if you are confusing it now, at least you know what you're supposed to do. For those people who are using this paper as a revision, okay. Now we continue. We are saying that state the phase of the state 1 place in human body where meiosis occurs. Meiosis occurs, we said the testis. We said in humans, we said it is testis. This is singular, this is plural. Or ovary. They said in human. They didn't say a male or female. So we go. Structure P is spindle fibers or spindle threads. Structure Q, we say it is centrioles. Yes, a centriole. Identify the phase of cell division that is being represented. I showed you, meaning that they are being represented by what? Diagram 2. Diagram 2 remember there is a space here. I told you to rotate it, so meaning that it is, since these are chromosomes, they are still together. Chromosomes, we talk about chromosomes, therefore it is, they are still 2, meaning that they are separating. These homologous chromosomes, they are separating. It means that separating apart, so it means that is anaphase. Since they are still 2, meaning that it is anaphase 1. Anaphase 1. Remember we said if they are still double, then we say 1. If they are single, then it is 2. You understand? We do the opposite, ne? Like this one, ne? They are chromatids. The chromosome is the one which separates. So it's single. Therefore it is anaphase 2 in diagram 2. In diagram 1. All right. So in diagram 2 is anaphase anaphase 2, anaphase 1, because their chromosomes are still double. And they're saying that how many chromosomes will be found in a somatic cell of this organism. They didn't say that this is a human cell. That's what I told you guys. Don't talk about human cell 46 chromosome when they say that. This diagram. So how many are they? 1, 2, 3, 4. In meiosis 1, those cells, those chromosomes are going to be 4. Why should you so talk about 46? Did I say that this is a human cell? No, I told you that. I hope you guys didn't disappoint me. So you deserve a trophy. Thank you so much, Mona Lisa. All right. Oh, I like that name, ne? Mona Lisa. Yeah, you know that Mona Lisa there. Yeah. All right. State one difference between the process occurring in diagram 1 and diagram 2. State one difference between the process occurring in diagram 1. Diagram 1, meaning that you're just gonna talk about here the chromosomes separate, they move to opposite poles. You understand? While here the chromatids move to opposite poles. That's it. Simple. This year, May, June, they did how do I say it? They need a trophy for you guys. You need to be getting good marks. At least you continue with your life. Most especially people who are writing May, June exam, these final exams. There are those people who are, maybe they have gone back, or they are upgrading. Maybe something did come up right, or they missed up the exam. They are not fresh. Those are people, it could be their first time, but their age is a little bit higher than those ones who are writing November, October, November. So you see, so thank you so much, the person who said. However much, yes, there are some questions which are not good for those who didn't prepare. But if you prepared with us, it means that you got it. All right. Chromosomes in diagram are genetically different due to they are genetically different. Chromosome in diagram are genetically different due to crossing over. All right. I told you don't go to the paper without knowing. Without knowing the process of crossing over, and we discussed it. Describe the process of crossing over. Talk about when does it occur. In prophase 1, what happens? Homologous chromosomes, they come together or they align. They exchange their chromatids. Yes, homologous chromatids at a point called chiasma. Yes, and then we say that after that, what happens? They, after exchange their genetic material, they separate in such a way that new genetic material from both is being shared. So basically this process, even I brought a diagram to explain to you. So guys, I hope you got this process right. You see such process, and then you get your mark. So yes, but those people who are doing the revision, you can take a screenshot. You pause it, take a screenshot. Now you know what you're supposed to write when they bring such. They're saying describe the non-disjunction. I explained this. How does it lead to Down syndrome? And I said, hey, when these are some of things which you might not miss out. Yes. So I said it here. Where are you? Yeah, after this. Yes, I explained it here. First of all, you need to explain to us non-disjunction. When does it occur? What happens? So basically, we say that you explain that it occurs in anaphase. yes, in anaphase. It can be anaphase 1 or anaphase 2, meaning that the failure of the chromosomes to separate. It tells us that the chromosomes fail to separate, yes, meaning that one result in one cell having an extra chromosome, meaning that it's going to have an extra chromosome. It's going to have 3 instead of what? Instead of 2. Or another one is going to have 1 instead of 2, meaning that one cell is going to have an extra chromosome, and then another one is going to have less of chromosome, meaning that it's going to have 23, another one is going to have 24, because it's supposed to be 23, another one is going to have 22. So if this 24 fertilizes a normal ovum or a normal gamete, yes, because we don't know what is this, a sperm or ovum, a normal gamete, of which is 23, is going to result in a cell, is going to result in a cell having, or an individual having 47 instead of 46. And if this non-disjunction occurred on chromosome number 21, therefore it becomes trisomy 21, which we call Down's syndrome. Yes, so that's what you're supposed to explain. I explained it. I can't repeat it now because take a screenshot. Because we're just discussing what are you supposed to be linked. Yes, I hope you guys, you got 10 out of 10. There we go. The next question 2. Question 2.3. The diagram below represents the stage of protein synthesis. Name the stage of protein synthesis there in diagram 1. In the diagram we say this is the moment you see these boxes. Remember I said that amino acids, they either circles or they are boxes. They brought them as boxes. So cool. State the process. We say this is the translation. Name the stage of protein synthesis. So we say this translation. Then they are saying that name the process. Name the stage of protein synthesis. Okay. State the site where the process named above occurs. It occurs on the ribosome in the cytoplasm. In most cases, they want to see the word ribosome. Sometimes they want to see cytoplasm, so that's why I told you that I prefer you to say that for you to say that ribosome in the cytoplasm. Definitely they will give you. If they want this one, they will give you. We have to be there. You said diagram 1. Someone was okay. I was just reading one of the comment. Diagram 1 opposite chromatid put opposite pole. Diagram 2 chromosomes are put opposite pole. Yeah, that's it. That's what I said. Yeah, that's what I said. Tabo, Mona Lisa. That's what I said. Cool. It's cool. All right. We go to the next question. Which question is that? Describe the role of X in protein synthesis. Role of X. What is the function of this in protein synthesis? In actual sense, they're asking you, what is the function of transferrin A in protein synthesis? It brings the required amino acid or the specific amino acid to the ribosome. Yes. So if you want to add, yes, when the anticodon of it matches with the codon of the messenger. When the codon of the messenger matches with anticodon of molecule X, yes, molecule X will bring the required amino acid to the ribosome. If they're asking you to describe that part, just to remove that part of what? Of translation. Yes, that's what they are looking for. So basically, this is what I'm talking about from our distinction material book. This is what I'm talking about. I told you that this book, I don't want to show you everything, but if I want to show you each step by step, you would have seen. So you see, this is what I'm saying. Transferrin A carries specific amino acid when the anticodon matches the codon of the messenger. Therefore, that molecule X will bring the required amino acid to the ribosome. So this is what I was trying to say. You see how many marks are there? There are a lot. All right, we continue. This identify the complementary base from left to right, representing anticodon 2. let me first clear it up, and then we see anticodon 2, anticodon 2, anticodon 2. So anticodon 2 is anticodon 2. We are seeing that CAU. They're saying identify the complementary base. Identify the complementary base from left to right, representing anticodon 2. So it's gonna be C goes with the G, A goes with the U, and U goes with A. So you get the mark there. They're saying on DNA codon 1. So I go to codon 1. Is if I pick this one, it's gonna be codon 1 AUA, AUA, AUA. So I need to convert it into DNA. A goes with T. Remember, this is DNA, and then U goes with A, and A goes with the T. Third. Table below represents the anticodon for different amino acid. The mutation, I told you this one is coming, is coming, is coming, is coming, is coming. Make sure that you do. Then they're saying that Maria, we have not finished the paper. Why do you say that you didn't? You failed it. Ayanda, I see you. All right. They're saying that a mutation occurred during replication. The mutation, I know that this one is gonna kill some people, and that's why I emphasized it. Make sure if you don't do it, you are gone, man. All right. They're saying that use the table to explain how this mutation will affect the protein. So we want to know whether the protein remain the same or is gonna change. But remember, you need to show us where did it change, which codon number or which anticodon, which code, all those things. Yes. But now they have given you, this time they have given you what we are supposed to get. So what they say is the mutation, what happened to the mutation? The mutation changed this to this. All right, let's find out. Prorine to arginine. So this one, what does it code? I'm just doing this so that you guys are able to get it right, né? And then what is arginine is CGU. What is this one doing? This one is gonna go back to CCU. So C goes with the G, and then C goes with the G, U goes with A, and then so this is the anticodon, ne? So this is codon. Because they say that use this explain how the mutation will affect the protein. The mutation occurred during replication. So replication, it means that it occurred on DNA. That's why you want to find out which code from DNA where the problem came from. Then this one, C goes with the G, G goes with the C, U goes with the A. So this is codon, meaning this is messenger. And then now let's go to DNA so that we know exactly what happened. They're saying that C, A goes with T because it is DNA. So it's gonna be G, C. It's gonna be C, G, T. You understand? So what happened if you look at it? The second base C was replaced with G on DNA during replication. So that's the mark. You have identified what happened. You understand? But they say 7, so it means that you have to show which is transcribed as GGA and GGA respectively on what? On messenger, and then CCU and G and CGU respectively on the transferrin A. That's anticodon. Hence proline and arginine, which are two different amino acids resulting in different protein. You see? So they wanted you to identify what happened here. They will give you a mark there. You translate it to messenger. They will give you a mark there. They will give you a mark there. And then yes, that codes for that. They will give you a mark there. And then they will give you a mark there. So they will keep giving you marks there. So you explain what happened. So that mutation, you always, if you see that the amino acids are different, it results in the same protein, sorry, in different protein. If the amino acids are the same at the end, result in different protein. All right, I hope you guys got it, yeah. We got the next question, that is question 2.4. They're saying that in some chicken, feather colors are controlled by alleles of alleles, feather, W and black. This is white, and then this is black, but look at the letters are both the same, meaning that they are dominant. So it might be related to incomplete dominance or codominance. If the heterozygous white chicken is crossed with the homozygous black chicken, offspring have the feather that is both black and white. Then they are saying that known as so when we are using the example, we use that one of flowers. Yes, we use that one of flowers, and then we explain it. Explain why this inheritance is an example of codominance, because it results in offspring having both colors, that is white and black and white, meaning that it's a mixture of the two colors. So I told you that is a mixture. It's a mixture of two colors, that is black and white. Then we got the next question. They're saying that explain why this the inheritance of blood group in human shows the same type of dominance. Explain. Yes, because when A is crossed, homozygous is crossed with B homozygous, they will result in AB, which is the codominance. That's it, meaning that A is dominant, B is dominant, and then you form AB, which is, it looks the same like this one, because white and black, then you produce a mixture of the two. You see that this looks like that. White chicken is crossed with the eminent chicken. Using the genetic cross, meaning the eminent is this, is this, yeah, cool. Use the genetic cross to show the phenotypes. This one is simple, guys. Remember I told you that when you have this, you have 3 marks free. So P1, F1. So you got, I think you got that mark before you go to the paper. You went to the paper, they said phenotype, we said genotype, we said meiosis, we said gametes, we said fertilization. So we say that you got this mark also. So those are 2 marks. And I say that for phenotype, you go to the question, they say that white chicken. So I said white chicken crossed with eminent chicken. That's another mark. You see, 3 marks. That's what I said. So you got it. So now they're saying that genotype, we say that white chicken, they said white is W, isn't it? Is W, so it's gonna be capital W, and eminent is capital W, capital B, because it's a mixture of the two. Then they say that meiosis, we say that capital W, capital B, then we say that capital W, capital B. Then these are gametes. Then we say this cross with this, capital W, capital W. You can use the Punnett square, capital W, capital B, correct, capital W, and then this with this, capital W, capital B. These are offsprings. What do they want? They say the phenotypes. So if they were looking for the phenotype, so I will come back here and I will say that if they're looking for phenotype, I have 1, 2 white chicken, and then 2 eminent chicken. Yes, 2 eminent chicken. So basically that's it. You got it. So they start to mark, but you went to that paper when you know they wanted to see this could be a compulsory mark, that if you didn't get it, instead of getting because there is a lot of marks here, there is a mark there, there is a mark there, there is a mark there, so there is a lot of marks there. All right. Then we got the next question, which is question 2.5. Question 2.5. They are saying that hypophosphatemia is a sex linked. They said it's a sex linked. You see, I said whenever they say sex linked, you use X and Y, inherited condition that is caused by dominant allele capital X and on X chromosome. They said dominant. You see, dominant. Yes, it is as a result of abnormal 2 level of phosphate. It's as a result of abnormal low levels of phosphate in the blood, which can cause rickets. They're saying that the recessive allele small h code for normal, meaning that if you have to be with both like this to be normal, or you have to be with this to be normal if you a male. All right, they're saying that male without. Okay, they give you give the type of diagram shown. How do you call this? We call it a pedigree diagram. They're saying gonosome found in male, gonosome found in male is Y. Yes, it's gonosome, meaning that the chromosome which is found in male. Yes, yeah, male only. They don't want you like this. They say male only, meaning they are looking for that chromosome which is Y. State 2 effect of this, they said it here. They said it here. It will cause rickets and also causes abnormal low level of phosphate in the blood. So they say that how many individuals in F1 generation have this condition. We say that the one which is shaded, remember this is shaded, this is shaded, the one which is shaded is suffering from the disease. They said in first F1 generation. This is F1 generation, but this got married, so meaning that it is this one, and this one, meaning there are 3 individuals in there. So 3 individuals. They're saying that all possible genotypes of individual 3. All possible genotypes of individual 3. Where is individual 3? Let me clear it up. We say that individual 3 is normal. And normal, they said that you're supposed to have the recessive allele, and this person is normal. So this is a male, so it's gonna be like that because he's a male. And then individual 4 is having the condition, meaning that is gonna be X capital H and then X small h. Why not X capital H, X capital H? Because they produced a person who is a female and is normal, meaning that this one is like that. So meaning that one came from the father, one came from the mother. So the mother can't be like this. That's why this is the only option which is there. So you get 1 mark there and you get 1 mark there. Explain why all the daughters in one will have the what? Will have this condition, and they say the 6 marks. So do you know what they want? They want you to cross, do a genetic cross, daughter. Yeah. So we are going to show you how are you going to explain. We say that the male, who is the male? The male is here. We say the male is what? Is sick, meaning that if the male is sick, it's gonna be capital H, meaning that the female is like this, isn't it? And then the female is like that. So when I cross, okay, after that you're gonna explain. When I cross, yes, okay, I have that and I have that, and then the female is gonna be small h, yes, you understand? So now I want to find out if all the daughters are going to be sick. Then from there we're gonna explain why. Then this gonna go like that, capital H, small h. Okay, that is a daughter is sick because it has a capital H. Then this gonna go with that, so it's gonna be capital H, yes, and then small h. Okay, this is also a daughter. Then now this with that is gonna be capital H, small h, and then y. This is normal. Then this one with that also gonna be capital H, small h, and then y. So you see the boys are normal, while the girls are sick. Not sick, they have that condition. You understand? Now, how are you going to put this in wordings? You understand? So when we come back here, we see that the boys are normal, you see, and then the females, all of them are sick. So what does it mean? It means that the males have a dominant allele. You understand? And then the females, they have 2 recessive allele. You understand? So the male will only transfer the Y chromosome to the males. You see, they will only transfer the Y chromosome, these Y chromosomes, to the males. Are we together? Meaning that all the X chromosome which are dominant, causing the individual to be sick, are going to be transferred to the females. You see, the ladies, the female, the mother, will only transfer the recessive allele, which will be masked by this dominant allele, causing all the females to be what? To be sick with that condition. Which condition is that? They said hypophosphatemia. Yes, I think now you get it, what I'm trying to say. Yes, I wrote 15 line, yo. Yes, it's fine, because you were just supposed to explain to us that the male have the dominant allele in this case, and then the female has 2 recessive alleles. The male, the Y chromosome is going to only be taken to the males, meaning that the dominant allele on the X is going to be transferred, or is going to be inherited to the female offsprings. You understand? The daughters. You understand? Yes, the recessive allele from the mother would be masked by the dominant allele, which was transferred, which was inherited from the father, making all the females to be having that condition of hypophosphatemia. Hey. All right. So someone saying, yeah, I get it. All right. Thank you. Since you got it, thank you. We go to the next question. Is just it was a dominant allele, and then I said that when they bring dominant allele, people go off. All right, we continue, guys. Thank you so much if you got it. And thank you for if you get it. If you have a challenge with the question, you tell me. All right, they're saying that describe the describe. They are saying describe the process of cloning. So what do you say when it comes to cloning? We talked about the process of cloning, and let me just use the distinction material book, and then just to show you what you are supposed to talk about when it comes to the process of what? The process of what? Cloning. So we said that we use a sheep, which we call the dolly sheep, but it's still the same process. Use the same what? You see the same process of cloning. It is. So it means that you have to first identify which cell are you going to use. You understand? Yes, you have to first identify the somatic cell. Get a somatic cell. We said get a somatic cell from an individual whose characters are needed. Yes, step number 2. Then remove the nucleus, and then keep the nucleus. Then step number 3, get another cell, a mature ovum from another sheep. Yes, and then after that, basically remove the nucleus and keep the cytoplasm. Yes, or the casing. And then after that, combine the nucleus of this and this and the cytoplasm of the cell from the other sheep, then with mild electric shock. Then after that, it will form a zygote like cell, which is allowed to undergo mitosis to form a ball of cell. So those ball of cell is gonna be split into small embryos, which are going to be inserted into the what we call the surrogate sheep, so that now the surrogate sheep can develop that embryo into a mature or a baby of that sheep. So basically that's what you're supposed to explain, link to this, and explain, said this is what we want, nothing else. Yes. And there's someone who said that you talked about it, but I neglected it because I knew that it's gonna come in objective. Hey guys, I'm sorry. We go to the next question. Name 3 benefits of cloning. Basically, the benefits of cloning, we can use it to get medicine. Yes, like this. Where is that? Yeah, to get medicine like this. Basically, they are looking for this. To get medicine. Where is it? Yeah, medicine. Yes, medication. Basically this is what I wanted to say. This cloning of insulin. Yes. We can also be used for producing identical organisms, which can be used for research purposes. Yes, process of cloning. Yes. And then we can also use basically what else can we use for cloning? We can use it for research. We can use to do the medication. Yes. And then we can use it to produce. There is this scenario also which occurs, like preserving the endangered species. Like if the species is about to get extinct or is about to get extinct, we can bring that genes and then we clone them, and then we come back, we bring them back to life, like so that we multiply them in large numbers instead of getting extinct, like one organism remaining or 2 organisms. So we can do the cloning so that we multiply them to be many. Yes. All right. Someone said doesn't know the process of cloning. I did a full video about cloning. So just go to YouTube, type cloning process, and with the word THUNDEREDUC, it will come. Yes. Those people who are using the distinction material book, this book, then it is being explained based on this distinction material book, meaning that at least, yes, you'll be able to use the same information. Let's go to question number 3. What are they saying? Question number 3, read the extract. Sickle backfish are small fish found in the ocean of the freshwater lake. The one in the ocean have spike fin for protection against predators until about 100 years ago. Most of the stickback fish live in the ocean, but the last ice age ended. The melting ice created new lakes and some population swam into the lake. The connection between the ocean and the lake eventually died up, trapping the stickleback population in the small lakes for thousands of years. Over time, the stickleback fish population in the lake lost the spike fin. Okay, how will Lamarck explain this? Remember, this is absence of this. How did they become with absence? Remember, Lamarck uses law of use and disuse and the inheritance of acquired characteristic. So what are they saying? You need to explain based on Lamarck. So you're gonna explain to us that there was no variation among these fish. All of them had what? Had the stickback or spike fin. All of them they had the spiky fins. Okay, that's step 1, meaning that there was no variation. All the fish they had the spike fins. So number 2, these fish, they stopped using their fins because there was no need for them to use. remember they were using them for what? They were using them spike fins for protection. They are using them for what? For protection. But where they went, there were no predators which were against them, so they stopped using their fins because there were no predators acting on them. So eventually the spike fins started to decrease and then eventually disappear. You understand? Yes. Then they passed this character of no fin to the next generation, and then all of a sudden, all fish in the fresh water they had no spike fins. Yes. So you explain based on what is being asked in that regard. Yes. So you link. You don't use giraffe. You can use the same concept, but you in this case you talk about the spike. And I said that last time, I said that they might bring it in the disuse. You have to use one. Either use it and then they develop, or you don't use it and disappears. Here you don't use it, then it disappeared. Why? Because there was no need for them to use the spike fins to protect themselves from predators, because where they were no what? There were no predators. Therefore they stopped using them, and then all of a sudden the fins started to decrease in size, and then they disappeared completely. They passed this character to the next generation. The next generations, they were born without the spike fins. That's Lamarck's theory. The diagram below represents the skull of pelvis of human and chimpanzee. No particular order. Cool. They're saying the name 3 structure differently. We did this several times. Okay, we say that draw a table, a complete table. Some of you did it just a T, 0. They're saying that is A and that is B. So they're saying A, you tell us. We use the same example. You people. We say that they have large canines, small canines, rectangular palates, U-shaped palates. Then you see the foramen magnum is in backward position. The foramen magnum is in forward position. What else do you want? You are shocked? Someone saying that I'm shocked I didn't see this question. Sorry. They're saying state Y pelvis C is regarded to humans. Remember we said that the one for human is short and wider. Short and wider. That's all. Guys, this is the things. I don't want to go there. This is the thing we said, and the emphasis said that, hey, when I don't make mistake to go there. Yes, I gave you that actual yes. Okay, we use this, but still I use the same thing. No, this is not what I want. This, yes, it is short and wider. It's the human. Remember I say, see, it's a human. This is the bipedalism for human. Biped, because we walk on 2 limbs. You see, it's a human. So it is the answer here they wanted is short and wide. That's all. Nothing else. Short and wide. Question 3.4. They are saying that malaria parasite, I talked about this also. Malaria parasite is transmitted to human blood by mosquitoes. People living in malaria affected areas take anti-malarial drugs to prevent the infection. If the malarial drugs are effective, there should be no parasite in their blood. The presence of malaria even after treatment indicates they have evolved to be drug resistant. Explain. The research wanted to determine if the use of anti-malarial parasite could causes the resistance in malaria. Okay, they gave you blood sample was collected from people of this affected area. Okay, the collection of blood sample was done in several villages. Okay, and the same blood were kept in the laboratory and tested. Then the number of sample having the malaria parasite was counted. They're saying investigation was repeated 3 times. That is good. They're saying that in total, okay, that is good. They're saying that was collected, okay. They're saying how did malaria parasite transmitted? They said through blood by the mosquitoes. So that is the answer. They're saying what is the independent variable? The independent variable is, they said it here. They say that the researcher wanted to determine the use of anti-malarial drug caused resistance, meaning that the independent is antimalarial drug. So that is the answer there. And then what depends is the drug resistance. Drug resistance will depend on which type of antimalarial drug they are using, or you can also say presence or absence of malaria. It can be there, presence or absence of malaria. They can also credit you, but you saying that resistance is a key point. Calculate the average drug resistance in the blood sample taken. Show your working around of your answer to. Okay, here you are saying that the percentage of drug resistance. They're saying that the sample were collected, 511 had malaria parasite, meaning that they were able to resist. This one they didn't have the parasite, meaning that I'm gonna say that 551 divided by 1485. 1485 times 100, which gives me 37.1. They said 1.1%. Name 2 ways in which you can increase or researcher ensured the following. Validity was done. Basically they use the same blood testing blood sample. They tested the blood sample from the same individuals in the village. Yes, they said it here, the same area. Yes, meaning that the same village. Okay, cool. Then they also use this the same testing method. They didn't change the methods they used. They said they tested it, so they used the same method. And then how did they ensure, because it is past tense, how did they ensure the reliability? They used the large sample size. Don't just say large sample size. They used 1,485 individuals or samples. 1,485 samples. Yes. Then number 2, that's the large sample size. They repeated the experiment. They repeated it three times in the same same village. That was reliability. That was validity. Yes. They repeated it. They repeated the investigation 3 times. Yes, that is also reliability. So you tell us what they have done. Yes. Then they are saying that describe how malaria parasite developed drug resistance. In this case, during our example, we used what you call DDT. Yes, we use this. With this DDT. How? So the first step, you need to talk about variation. The second step, you need to talk about selective pressure. The third step, you need to talk about the interference. The fourth step, you need to talk inheritance. The fourth step, you need to talk about the adaptation, meaning that in this case you tell us that there was variation among the parasite to the there was variation among the malaria parasite, those which are resistant to the drug, and then those which are not resistant to the drug. Yes. When the first application is done, yes, what happened? Those which are not resistant, they died in large numbers, while those which were resistant, they survived, but those were few numbers. You understand? Yes. Then those which survived, they pass their genes to the next generation. You understand? After many generations, yes, the resistant one, they increased in the population. You understand? Resulting in the resistant parasite. Therefore, the drug will become ineffective, hence resistance. So basically, you explain exactly this. Instead of talking about DDT, you talk about that malaria parasite. Instead of talking about DDT, you talk about the malaria parasite. That's it. That's what we call the application. The question, when the question comes, you just replace it. So that's what they wanted about these mosquitoes. Yes. So yes, then the last question, this one we did it. I don't know how many times we did it, but we did it. And I told you that don't make a mistake to go to the paper when you don't have this. This is what I told you. Don't go to the paper without knowing these questions. And I went through them, and you'll find out that all the questions they said they are there. And they're saying that number one, how many genera are there? Yes, how many genera are there? How many genera are shown? You see, that's the question. And then they're saying how many genera are there. So you look at Ardipithecus and Homo. So there are 2. So basically 2. Because this is Homo erectus, Homo neanderthalensis, this is Homo sapiens. All of them are Homo. When did the Australopithecus africanus became extinct? The question is there. When did the Homo habilis became extinct? You see, they're saying that. So Homo habilis, they say now Homo Australopithecus africanus. So it's the same thing. So it stopped here, meaning it's 2.0 million years ago. Then they are saying that name 2 Australopithecus africanus species in South Africa. Name 2 Australopithecus africanus species in South Africa. So this is what I told you, that we always ask you that when they come to the fossil, they're going to ask you name 2 Australopithecus africanus species, you know. So you can talk about Taumchild, Mrs. Place, or Littlefoot. They said africanus, Australopithecus africanus. Yes. So when you come back to that, yes, is this thing Australopithecus africanus species. That's when I was explaining it, I said this. They are saying that they use only the species in the diagram describe the fossil evidence, meaning the species only. Remember we said that Australopithecus africanus, you give example, yes, and Homo habilis. Australopithecus africanus species only were discovered only in South Africa. And then here Australopithecus africanus species only in South Africa. And then Homo erectus, even this one, Homo neanderthalensis and the Homo sapiens, the oldest was discovered in Africa. This is what I'm trying to say. Yes, because based on the question, they only want, you see, this Ardipithecus, they didn't talk about it. They started from here. Australopithecus africanus, yes, and Homo habilis only found in Africa. Yes. And then the oldest fossil of Homo erectus and Homo sapien were found in Africa. I said that this one, you must use the word oldest. If you don't use the word oldest, you are gone. Yes, so, and I repeated it, guys, and I said make sure you write the word only, because if you don't say the word only, then you are gone. They're gonna cross it. So, and this word oldest, it must be there. Don't make a mistake to do that. That's what I emphasized you, and I begged you. I said that, guys, this question, when it comes, make sure that you answer it properly. So guys, those who are revising with this paper, make sure that you use it now the next time they bring it. But still, in the next paper, we'll also predict for you, and then we think what is gonna be coming in that regard. Explain how the influence of brain volume on the complexity of the tools made by Homo habilis and Homo sapiens. How is the brain size? This question is linked to the question, which I asked you, that how is the brain size? This one, the last one, how is the brain size? Describe how the brain size evolution trend. How does the brain size show the evolutionary trend? This is the question which I said that make sure you answer those questions, guys. Yes. Explain how is the brain volume, meaning that the bigger the brain volume, the bigger the brain, yes, the higher the number of brain cells, meaning that the higher the intelligence. You understand? They're saying that the complexity of the tool, you understand? Basically, it shows that, as actually I showed you this diagram when I was explaining it. I showed you this diagram Where is it? Yes, this diagram. And I say that as the brain size increases, yes, the number of brain cells also increases, meaning that the intelligence increases, meaning that as also the tools also became more advanced. So the brain size, as the brain size increases, the advancement of tools also increases. So as the brain size increases, okay, the advancement of the tools also increases. Then why? Because these tools require thought to be developed. You understand? Yes, that's what we said. And then, yeah. And after that, I told you guys, I wish you the best. And then, it's the same conclusion. See that, yeah, it was 99% success prediction. Yes, and we will always predict for you, and we want you to pass. Stay with us, study from the beginning until the end. I promise you, you will never regret you studying with us. There are some people who try to study with us, they put some pressure or say, no, we know what we are doing. Yeah, we know, yeah, what we are doing. Yes, and we know that step by step. You don't need to rush for something before you grasp and understand the concept. Thank you so much, guys. Thank you. Thank you all the people. Sabero, I see you. No cross night for all of it, saying that geography paper 2 was released already. I didn't do a lot of useless mistakes, I'm sorry. So thank you guys, thank you, I see you, and I wish you the best in your life. And if things don't go right the way you wanted them, you can register again. You can register again by, I think, by what? By what? By August. Yes, and then you never know, you might make it again. Thank you so much. See you again in the next class.